Okay, well, thank you very much, Chris. I do really appreciate the opportunity to talk to everybody today, and I thank everyone for tuning in to hear the presentation. So, yeah, what I'm going to talk about is some work that we began in 2023, actually. So this is our second year of doing this work. We're a group at the Illinois Natural History Survey that has been focusing on monitoring for a long time now. So most of us are part of something that's referred to as the Critical Trends Assessment Program, where we've been monitoring forests, grasslands, and wetlands throughout the state since 1997. And yeah, this is kind of a shift in a slightly new direction for us, and I'm pleased to be able to tell you guys about it. Okay, so um, problems associated with pesticides are certainly not anything new when we're thinking about the natural environment and natural ecosystems. A lot of us, when we think about pesticides in natural ecosystems, think about largely problems that started popping up post World War II when synthetic pesticides became much more popular. So thinking about things like DDT and dieldrin and how we over time started to learn some of the negative consequences of those types of chemicals. I think what a lot of people don't realize is that the increase in use of pesticides is something that is still going on today. So looking at this graph here, this is a graph of global pesticide use since 1990, and you can see even in the past 3 decades, we've seen pretty remarkable increases in use of pesticides, and a lot of it is driven by actually herbicides and then to a lesser degree fungicides rather than insecticides. So that's a global look, and if we look at Illinois specifically for the most recent years that we have data, you can see that yeah, indeed, um, there's lots of things that have been increasing pretty dramatically, including lots of herbicides that have been around a long time that are increasingly being used, including some things like dicamba that have been technically around for a long time, but have been starting to be increasingly used more. And then things I think a lot of us have heard about things like neonicotinoid insecticides, which are being used more frequently as well. The graph on the lower right being a little bit misleading because the USGS stopped tracking to some extent around the year 2014, 2015, because most of these are applied as coatings on seeds rather than directly applied as a liquid form or granular kind of thing. Um, and the thing that has increased awareness of a lot of people has been since 2017, 2018 or so, increased focus on dicamba. So, development of genetically modified soybeans, for example, to be able to overspray dicamba happened around that time. The reason for this is that herbicide resistance is increasingly becoming a problem. So additional tools are being developed. People are looking back towards chemicals like dicamba has been around since at least the 1970s. But it is something that we've known, at least since 1979, that dicamba, for example, is quite volatile. So there's been a resistance to go to using that, but the problems associated with herbicide resistance in weeds has led us to start looking at those things again. So with that, we've started to see some increase in reports of damage and things like that, and that's what I'm going to get into a little bit more. Likewise, as I've alluded to on a previous slide, things like neonicotinoid insecticides have increasingly been a focus, and they're ubiquitously used as seed coatings along with fungicides and other things on corn that's planted in Illinois, and a large proportion of soybeans as well. So these are things that are in the environment as well, and we don't necessarily have a good handle for how widely these things are moving in the natural ecosystems. Um, Going back to herbicides specifically, with that increased use of dicamba starting around 2017, we started seeing a really big spike in the complaints, a lot of which was driven by the agricultural community. So non-target effects, for example, on soybeans. So some people, for example, spraying this relatively volatile herbicide on soybeans that are genetically modified to tolerate it. Lots of individuals being out there that don't have those genetically modified soybeans starting to see damage because of the volatility. So we saw a spike in those numbers around 2019, and since then the complaints have kind of tapered off a little bit. And to some extent, in Illinois, and this is true in other states as well, we've seen some sorts of responses at state levels in terms of trying to set some kind of boundaries in terms of application dates and conditions, for example, under which this chemical could be applied. That gives us a sense of kind of what's been going on in agricultural fields. But at the same time, around 2018 in particular, there's been this increased interest in like, well, what's going on in natural areas? What's the potential influence of these chemicals making their way into things like nature preserves, into, state parks and, other types of natural areas, including, privately owned forests, things like that. And over the past several years, we've increasingly started to see evidence of this showing up, and we've seen a little bit of research, but we've seen, a fair number of news articles, for example, of people starting to recognize that this, in fact, is a problem. In the state of Illinois, the Rivers Network is a group that has been looking into this since, I think, 2018 or so, and they've been documenting damage in a lot of different areas. And, it was based on the understanding, that this is potentially a wider spread problem, largely things that they have documented, and they did that in this nice report that's out there and available online. That we were contacted to start working on this problem as well. In the context that we were approached about working on the problem was related to, first of all, just kind of understanding the extent to which this is a problem. A lot of the interest was in nature preserves specifically. So the Illinois Nature Preserves Commission is responsible for ensuring the protection of lands that are listed under that program. So that includes dedicated nature preserves and land and water reserves, and then one other class of site as well. Those sites have special legal protections under state law. So, we kind of approach this from just trying to understand, well, what is the extent of damage that's out there? What are the species that appear to be most affected by this potential herbicide drift problem? Likewise, just a better understanding of what chemicals might be responsible for this. So, dicamba, I guess, is kind of the starting point for a lot of people, given the way this problem kind of evolved over time. But we wanted to take kind of a broader approach of just to look broadly to see, well, what kinds of chemicals are out there, including herbicides, including fungicides, insecticides, and other types of things as well. Likewise, what are the predictors of where this is more likely to occur? So are we seeing particular areas of the state that are more heavily impacted than others? Are there certain types of natural areas that tend to be a little bit more susceptible than others? And to what extent can we predict that? Likewise, given that we were interested in the special class of INPC, Illinois Nature Preserve sites, what is the status of nature preserves versus other types of natural areas throughout the state in terms of the degree of potential damage and pesticide exposure that we see in these? Likewise, how do these risks change throughout the growing season? So as you might be able to guess, with agricultural activities, chemicals are applied at different times of the year. So you might expect to see damage or be able to detect pesticides differently at different times of year. And we wanted to get a better understanding of that. The way we did this was with a random sampling approach. So, in contrast to some past approaches that were focused on, getting a report of we've seen damage at a particular site, let's go to that site and sample it and confirm damage and that kind of thing. We just started with a completely random sampling frame. And what we really were interested in is just this broad assessment of pesticides and plant damage throughout the state, in natural areas, including, public and private lands, nature preserves, and other types of areas as well. So the way we did this, again, was starting in 2023. We focused on 185 total sites. 83 of these were completely randomly selected. These were originally sampled as part of our critical trends assessment program. Some of these sites have been sampled since 1997, where we're trying to get a sense of kind of the natural community and changes over time in those natural communities. And then onto that, we added a spatially random sampling design for nature preserve sites as well, including land and water reserves and nature preserves. So again, trying to make sure we have good coverage throughout the state, that we get all the different contexts in which these sites occur and get a better understanding of what things might lead to greater risk. As I alluded to before, we were interested in that kind of seasonal component, so we did 2 different visits per site. Um, one of these is the early growing season, so between May 17th and July 6th. Our second sampling window started on July 17th and ended in early September. Okay, so what did we do out there with respect to trying to understand what chemicals are out there? We did tissue sampling, so we were collecting leaf tissue. We were both collecting tissue from plants that clearly seem to be showing some signs of damage, but then we also picked completely random plants as well to try and get an understanding if there's any difference there. All of these sites were greater than 20 meters from an edge, so we weren't interested in just only kind of capturing kind of these edge types effects where there's known to be potential problems there. And then we also focused on collecting a soil sample as well to understand if there's any kind of accumulation of chemicals in the soil. All of these samples were sent off to a commercial laboratory for analysis. When I say that, we were looking for 486 total potential pesticides or pesticide analytes and metabolites. So a pretty broad list. Again, we weren't wanting to assume that it was one particular chemical, wanting to get a better sense of kind of what was out there. Paired with that, we were doing these sampling of the natural communities, these timed meanders done by trained staff, many of which are botanists. 10 to 30 minutes per visit. There's a plant injury scale. It's done on a species-specific basis. So, particular species, what's the extent of damage on that scale of 0 to 6? It was a process that was standardized across observers based on training, but then we were also able to control for observers using statistical types of approaches as well. On the right, you can see kind of a sense of what this data sheet looks like. So there are specific things we're looking at. The things in bold there are kind of indicative of herbicide damage specifically, whereas the stress-related things that aren't in bold could be related to pesticides, they could be related to disease, they could be related to drought or lots of other kinds of things that cause stress to plants. In addition, we were doing some marking of individuals so that we can hopefully track these over time. So that included, over 1,000 individual plants, largely focusing on trees that we marked, measuring the DBH, recording diameter, breast height, recording the signs of damage in that particular individual. And then we've been returning to some of those this growing season and hope to continue doing that into the future as well. All right, so what did we find? So first of all, we of course did find pesticides in these natural areas. This gives you a sense for that early sampling window, that early growing season sample. These are the most prevalent chemicals we saw. So atrazine and then the diacetylform, which is a metabolite or degradation product of atrazine, were by far the most common. 2,4-D was also quite common at more than half of our sites. And then you see a range of other things that show up occasionally, metolachlor, glyphosate, a few fungicides showing up, and you see dicamba showing up, but at a relatively small number of sites throughout the state. In contrast, when we look at that second sampling window that started in late July, you see that we have the shift to largely fungicides and a few insecticides being the most prevalent thing out there. You see atrazine and 2,4-D still there. They've kind of shifted down a little bit because these things just naturally break down over time. They're applied early in the season and just kind of degrade a little bit. But applications of these fungicides and insecticides happen later in the year. In total, we had 38 different chemicals that we detected in leaf tissues or in soils. In terms of just numbers that we found in samples, so at least 74% of our leaf tissue samples had some pesticides detected in them. That included 97% of our sites. On average, a leaf tissue sample had about 2 pesticides on it, but that could range from none to up to 11. On the right side is listing the things that we did find out there. Herbicides, again, tended to be the most common, followed by fungicides, and then followed by insecticides. So there are a broad range of chemicals that are making it into these natural areas. Again, focusing on that kind of temporal component, the blue and orange lines here correspond to atrazine and that metabolite added together, as well as 2,4-D. And you can see, as you might expect, these things are applied early in the growing season associated with planting. Those kind of degrade over time, and then we see that big spike in a whole broad range of fungicides and insecticides that start showing up later in the year. The things that we did not find much of, so, as I mentioned, dicamba didn't show up very often at all. This map on the right shows us 5 sites where dicamba did show up, all in the same couple of days, interestingly. Neonicotinoids did not show up very often either. So, we know those are very widely applied on as seed coatings. This is suggesting that, encouragingly, it's not making it very far into the natural areas, although we do know that it ends up in waterways at large levels. In terms of soil contaminants, so we collected soil samples from all these sites. We sent our first batch in of maybe 60 samples or so, and we did not find much in terms of soil contaminants, so that was encouraging as well. We also didn't find anything in terms of statistical differences between our INPC sites, so like our nature preserves, and those randomly selected sites, which could have been, a range of different things, including state parks and privately owned areas and so on. So suggesting that this is a broad problem. It's not unique to nature preserves. It's happening at natural areas throughout the state. In terms of what predicts the concentrations of these chemicals, not surprisingly, you see in each of these graphs. So if we start by focusing on the graph in the upper left, that's atrazine and its metabolite as a function of the proportion of corn in the surrounding 1 kilometer. When we focus on that first visit, again, that's when we see those herbicides being most prevalent. You see, as the amount of corn in that surrounding kilometer increases, the concentration of atrazine increases as well. And this makes sense because atrazine is a primarily corn applied herbicide. When we look at, insecticides summed up and fungicides summed up, again, this is primarily happening during that second visit. So that's why, we're focusing on this red line. And again, if you look at the proportion of corn, you see that increase. It's notable that there's a different scale here, however. So in this case, we're looking at the proportion of corn in the surrounding 100 meters, whereas before with atrazine, we were looking at the surrounding kilometer. So this is true with both insecticides and fungicides, that it seems like it's a smaller scale at which that's operating. So if you've got corn within 100 meters, you're much more likely to get those fungicides and insecticides showing up. We did see some signal that soybeans were more associated with 2,4-D concentrations. And, in talking to people, it seems like there's actually been a decrease in use of dicamba over time, and it's largely been replaced with use of 2,4-D, and a lot of that has focused on soybeans specifically. This, was a pattern that was there, but it was not as strong as what we saw with corn. And as a general rule, for just pesticides in general, what we see is, the more row crops we have in the surrounding landscape, the higher the pesticide concentrations we see. So in this case is the inverse of that. So if we add up all the natural types of areas, so forests, grasslands, wetlands, shrublands, those kinds of things in the surrounding kilometer, as that amount goes up, the pesticide concentrations tend to go down. So that gives us a sense of just kind of the general context and how that influences what chemicals might show up. We can also look at this spatially and do a statistical approach known as kriging to do some spatial interpolation. And based on that, you can see that there are some kind of hot spots that you see for some of these chemicals. So atrazine, tends to be found in a couple of hot spots in the northern part of the state. 2,4-D tends to be found at higher concentrations and kind of a hot spot in the southern part of the state, although both chemicals are found statewide as well. So that's notable to point out. Likewise, when we focus on some of these fungicides and insecticides that start occurring in the later part of the growing season, propiconazole is a fungicide and bifenthrin is an insecticide. You see the same general pattern where some of them are showing up in much higher concentrations in kind of a key area in the north central part of the state. So yeah, indicating there is some kind of spatial predictability to where we're finding this stuff. Okay, so that gives us kind of an overview of what we see in terms of the actual chemicals. What did we see in terms of the plants and how they might be responding to some of these chemicals? So as I mentioned, these meanders were kind of a species-specific assessment of potential damage. Our observers looked at 78,000 total plants during the 2023 growing season at, around 400 total species. And 99% of sites had some sort of damage that was consistent with herbicides. Again, as I showed you that data sheet earlier on, you had certain things in bold that were kind of indicative of herbicide damage and some things that are more broadly indicative of stress in general. So yeah, pretty good evidence that there's some damage going on. Just to give you a sense of what some of this looks like, this is a red oak with showing some very characteristic cupping signs that's associated with herbicide damage. This is an example of a bur oak showing signs of damage, and you can see that both in the foreground on this part, and even in the background, you can see kind of some very unnatural kind of clumping of leaves that can be associated with the herbicide damage as well. Example of shingle oak, twisted and deformed leaves again, especially in this kind of area, so fairly widespread. Suppressed and deformed growth, in this case on a black oak. Strapping, which leads to this elongation, this unnatural elongation on a bur oak. And it's notable that, we're only able to see a certain amount of the potential damage that's out there. And a lot of it, when we're talking about forests in particular, is actually occurring at high levels, potentially in the mid or high canopy of trees. So, observers had binoculars out there to try and get a look at this. And, sometimes you can see it, but it's hard to get a good picture of it. But you see things like this that are showing up out there. Sometimes you see plants displaying multiple issues at the same time. As you can see, there's some definite growth problems going on multiple leaves in this particular case of this swamp white oak. Twisted leaves, deformed growth. This looks like some hickory species. Another deformed growth, this time in a box elder. So it wasn't exclusively oaks. A grapevine showing kind of very characteristic signs in terms of venation problems and kind of weird things going on at the margins. Another case where you can see this kind of death of tissue, but then you have new growth that's kind of occurring there as well. And I've been showing you a lot of trees, for example, but this was true in herbaceous species as well. So this is rosinweed on the left. This is a gray-headed coneflower that's showing signs of damage as well. So a fairly widespread amount of damage in natural areas in general. In terms of trying to quantify the species that are most affected, so we focused on species that we had at least 10 observations for, and this is the list of kind of the most affected species there. And one thing I think you'll notice is oaks are showing up very prominently on this list. So most of the things listed here are oaks. You do see things like redbud, you see things like box elder, hackberry, sugarberry, persimmon, a hickory showing up here as well. But oaks seem like they're really affected. There are some species, that were less frequently observed that also had very notable entry as well. So again, we looked at like 400 species, and there's a lot of them out there that show some kind of damage. As we did before, we can start asking questions about, well, what's the spatial patterning of this damage? And you can do the same kind of spatial interpolation and get kind of these heat maps to say, okay, well, there's certain areas of the state where damage tends to be much more observed. But we can also do the same thing where we look at the surrounding landscape context and get a sense of what's most associated with damage. And again, we see as the proportion of soybeans in the surrounding 1 kilometer increases, our overall site injury score tends to increase as well. And this is accounting for variation among observers, kind of controlling for that, which was not all that great in the first place. So observers were pretty consistent. Likewise, with the increase in corn in the surrounding landscape, you see the same type of increase in the site injury score. And then if you have more forest in the surrounding landscape, you see a decrease in that site injury score. So row crops really driving these patterns in particular. Also notably not much association with development, but some kind of weak association that as there's more developed area, you actually see decreased injury. So it really does appear to be like a row crop related problem. Um, the types of injury levels that we saw at our nature preserve sites, so nature preserves and land and water reserves, on average was pretty similar to what we saw at our sites that were completely randomly selected, that were part of our critical trends assessment program. Um, yeah, some level of injury at 99% of site visits, so very widespread. There's no real difference in kind of the average amount, but one thing that you can note looking at this box plot on the right, that our INPC sites had a greater overall variability. So you have greater extremes in these kinds of sites. So it includes some of the most negatively affected sites and some of the least negatively affected sites as well. And if you know anything about these types of sites, it kind of makes sense, because some of them tend to be very small, very isolated, and surrounded by agriculture, for example, whereas others are quite large and surrounded by more natural cover types. So sites with the greatest injury tend to be INPC sites. We also tend to see more damage in wetlands or grasslands. That's not to say forests didn't have damage, because damage was quite widespread in forests. About 50% of our sites overall had signs of relatively severe damage, actually. And agriculture was the primary driver, and we saw a somewhat stronger association with soybeans in terms of that plant injury score. Um, and so just to kind of review, the basic findings, we found pesticides and injury at nearly all sites. So, 97+%. We see this general pattern of herbicides early, atrazine, as well as 2,4-D primarily, a wide range of fungicides showing up late. So that's a general pattern. We see that, it really is associated with the amount of row crop in the surrounding landscape. The scale at which that's happening appears to vary a bit, depending on which chemical we're talking about, but it is really a row crop related phenomenon. We do see some hot spots of injury as well. So there's certain parts of the state where injury is much more likely to show up. Some of that may just be related to the general, landscape composition in terms of row crops and whatnot, but it's not completely related to that. So there's some potential other things going on as well. Um, the damage we were seeing was similar among the types of sites that we were looking at. So it does suggest that this is not specific to a particular type of sites. It's a larger problem that's going on at natural areas across the state. And then, additionally, the injury was worse in oaks. So that's potentially troubling for a number of reasons, which I'll get into here in a second. So this is work that's ongoing. So we've continued sampling in 2024 as well, returning to a lot of those same sites to try and get a sense of how much things vary across years, for example. But there's a number of additional questions that we're trying to get at as well. So, is there an effect on growth and survival in affected species? There's this larger question of, like, we've got a better sense of what's going on out there, but what are the implications of it, right? So you see some negative impacts in terms of the appearance of these trees, but does it have long-term impacts? So, does a single exposure event have a negative impact on the survival of growth? What about repeated impacts? So there's a bunch of those kinds of questions that we need to get a better sense of. So in general, at these more contaminated sites, so for example, the smaller sites that are more completely surrounded by row crops, do we tend to see slower growth and decreased survival? So those are some things that we're interested in understanding. The vulnerability of sites across year is another good question. We do, at least anecdotally from our sampling in 2024, see some differences. So we're still seeing very widespread damage in 2024. But there's some suggestions that in some cases it might be less severe. And at this point, we don't necessarily know the reasons for that, but the weather every year brings different things. So, it could be drier conditions last year, made plants more stressed. It could be drier conditions led to earlier planting and, a more, coinciding of the application timing with the periods of time when plants are more vulnerable during the leaf emergence and early growth stages, for example. So trying to get a better sense of those kinds of things, whereas planting this year was, tended to be a little bit later. It seemed like it was a little bit wetter. Maybe that has some kind of influence. Perhaps we'll find out. Likewise, some of these chemicals are more associated with corn. Some seem to be a little bit more associated with soybeans. As we see our typical crop rotations from corn to soy in most areas, we would expect to see kind of an alternating being hit by different chemicals each year potentially. So that's something that we're looking at in a bit more detail, although are some parts of the state, and that north central part in particular, where it seems like continuous corn may be more common, and that's actually potentially the reason why they're applying more of these fungicides and insecticides, because of problems associated with not doing those rotations. Likewise, we don't have any good feeling for, what are long-term effects at this point in time. It's really hard to understand, especially for things like trees that are very long-lived. If something's hit year after year, what kind of impact is that going to have? So we can start to get a handle on that. Although, directly observing these marked trees, it's going to take a while to fully understand that, for example. We are engaging in some greenhouse and lab-based experiments. So some of which is trying to understand what's the importance of exposure timing. So, by being able to control the environment and the concentrations that they're exposed to, hopefully we can get a better sense of that. Likewise, it's not a single chemical lots of times that they're being exposed to. On average, yeah, we're able to detect multiple chemicals on each leaf tissue sample. So what are the implications of being hit with multiple chemicals at the same time, multiple chemicals in succession, getting hit multiple times throughout a growing season? Those types of things are things that we want to understand as well. Likewise, what are the between year effects? So, we've got seedlings in the greenhouse that we expose this year, for example. We'll let those go dormant, and then when they emerge and leaf out again next year, we'll get some better sense of, are there persistent, longer lasting impacts that we're still able to see in year 2. So that's something that we really don't understand at this point in time. There are cases where we don't really understand how long the chemicals are persisting in plants. So we have some sense of the half-lives of these chemicals based on, work that's been done in agricultural systems in particular. But there's a lot of variation depending on environmental conditions, depending on the species. It's a different value for soil versus in leaf tissue. It can be species specific, those kinds of things. How long are the symptoms evident? So does it last across years, or when we see damage in multiple years, is that always the case of it getting re-exposed year after year? So yeah, some of those kinds of things. And then obviously there's a whole range of questions related to what kinds of impacts this might have on other organisms in a particular ecosystem. So, we're focusing pretty heavily on the plants at this point, but there's a lot of potential negative impacts on insects as well. So we've started some preliminary work focused on feeding caterpillars treated food that's either treated with control of water, or it's treated with atrazine or 2,4-D at this point in time, just to see what happens. It's definitely true for all these chemicals that they go through a screening process to be registered and re-registered. There's a focus on acute lethal effects, for example, the concentrations or doses that are required to kill 50% of individuals. That's a lot of it is focused on certain model focal species. So, for birds, for example, that I primarily work on, quail and mallards tend to be two of the model species that they use for those acute lethal kinds of impacts. For insects, the only model species is typically honeybee. Something that's a little bit problematic with that is, honeybees aren't, actually consuming the leaf tissue out there. And if it's the leaf tissue that has these chemicals on it, there's potential implications for the things that are consuming it. A thing that makes this a little bit more troubling. So as I mentioned, oaks were the most impacted group that we saw in our areas that we sampled in 2023. Oaks are also, the plant group that is most important for caterpillars. So primarily moths. And moth larvae are very important to lots of migrating and breeding birds, for example. So there's some potential connections there that could be a little bit troubling. So that's one reason why we started on this kind of lab-based approach. And one of the things we found in our preliminary work is that when we, treat their cabbage or kale with, control, just spraying them with water, they they reach pupation at a fairly high rate, and then subsequently they emerge from pupation at a very high rate. But if their food is treated with either atrazine or 2,4-D, we see effects on development. So development's a lot slower. They gain mass at a lower rate. They're much less likely to make it to pupation and much less likely to emerge as adult moths as well. So potentially negative impacts on insects that we don't fully understand at this point in time, and that may go on to affect other trophic levels as well. As I mentioned, a lot of these sites are areas that we've been sampling for a lot of years already as part of the critical trends assessment program. So we have a lot of data on some of these sites already. And now that we know attributes of sites that are most likely to be impacted, we're able to go back to those data and look at, for example, what is the potential influence on tree mortality rates. So tree mortality rates, pretty difficult thing to focus on given how long lived they are. But from 25 years of data or so, we're able to see that there are certain parts of the state where tree mortality tends to be a lot larger than in other areas. So we're trying to get a better handle on this kind of aspect as well. Likewise, we've measured these trees, each time they get sampled on like a 5-year rotation. So to some extent, we'll be able to look at things like growth rates as well. Do we actually see impacts on growth rates, and are those effects on growth rates or mortality species specific? So is it true that certain species like oaks or box elders are more affected than others? Likewise, this is just a question. This isn't something we're specifically working on, but there's a wide range of plants that are dependent on fungi for symbiotic relationships that help them out. The exposure to fungicides might have some implications for affecting growth in some of these kinds of plants as well. So that's something that we'll be interested in looking at. Are we seeing patterns in these species that we know have these associations? Are they tending to decline a little bit more than others? Fungicides. So, we focus a lot on herbicides. The potential damage associated with that is much more apparent, of course. We didn't necessarily, we were kind of taking a broad approach of trying to see what kind of chemicals were out there. And indeed, there are a lot of fungicides showing up, and a lot of these fungicides are relatively new on the scene in terms of, being applied in crop kinds of situations, and the use is increasing and increasing. We don't fully understand, what all the implications of this are in terms of how it might affect natural communities. So I gave one example of plants that are dependent on fungi. Decomposition processes, which are important for creating snags for certain species of wildlife. Woodpeckers, for example, is something to think about. Effects on, mushrooms. There's evidence that fungicides can have these synergistic impacts with insecticides that actually increase the toxicity of insecticides. So there's some of those kinds of things to think about as well. And then lastly, just trying to understand more about the mechanisms by which this is happening. So, we see kind of species specific patterns. So, we need to get a better handle on why that is, right? Is it things, just biologically inherent to oaks, for example, that makes them more susceptible, or is there something about leaf emergence that is making the window of exposure just happen to line up with when chemicals are more likely to be applied? So those are things that we're trying to get a better handle on as well. Likewise, very relevant to a forest environment, seeing variation across strata. So yeah, it's not uncommon to see this damage in canopies. So what's the mechanism for that influence? To what degree is it volatilization of some of these chemicals, which dicamba certainly does, although that was not very common in our samples. 2,4-D can do this as well, versus just like physical drift types of processes. So trying to get a better handle on that. And as I mentioned before, trying to get a sense of how things vary across years and how we can use that information to better understand kind of the exposure windows and how this influences plants, and what information we can provide that might help ease some of these problems. That's all I have in terms of information to present. I do want to thank everybody that made the work possible. So this was funded by the Illinois Department of Natural Resources, and we got a lot of help from INPC staff and other field staff within IDNR. Lots of help from great field technicians and folks at the Prairie Rivers Network helped us kind of get started in terms of how to understand how to do some of the sampling with respect to plant damage and so on. So I will stop there, and I would be glad to take questions. And as Chris said, yeah, just go ahead and drop those in the chat, and Chris will go ahead and read them out.